You're listening to the FaceTime with Scientists podcast, where we go into the lab or into the field to learn from scientists about their lives, their work, their struggles, and their joy. Let's go. Well, hello, everyone. I'm super excited to chat with Caterra Leavens today. You are at the University of Wisconsin-Madison, which I've actually worked with some science communicators in there. I know that Wisconsin-Madison has a big science communication, like health communication program. But anyway, very cool. I have seen your work on Instagram, some of your science communication there, and I've just been excited to chat with you ever since. So, yay.
Oh, thank you.
Can you tell us, a little bit what you're doing in the lab today? And
Yeah, thanks so much for having me. I'm excited to talk with you about the cool and exciting science we're doing here in my lab. And today, while we talk, I'm going to be doing some flow cytometry. And basically, what that is I make insulin producing cells, and it takes me a month to make those, right? And at the end, how do I know I actually have insulin producing cells? And that's where flow cytometry comes in, where I can actually look at gene expression of those cells to really ensure that I have the cell types that I anticipated at the end.
very cool. So, you have to remind me, I've done some cellular work. So, flow
Yeah.
cytometry is basically, you're sending cells in individually through kind of like a little tiny channel, and you're picking up signals from the individual cells. From that, I'm guessing you have them labeled for if they're
Yeah. So exactly, what I do is I stain my cells with antibodies, and I like to think of antibodies like little tags for specific genes. So in our case, like we're looking for insulin, and so I'll tag the insulin gene if those cells have it, and then I will send it through a little tube, single cells, one by one, and it'll if they have it, it will reflect that signal that I tagged them with. And if they don't, well then I don't have the right cell type, and so it's kind of instant gratification, which I love about flow cytometry as well. Instead of taking a month to do something, I can analyze cell lines easily and within a day.
that's awesome.
Yeah.
Very cool. So, yeah, what does it look like when you're doing this? Yeah, can you talk us through a little bit about,
Yeah.
like, the prep?
Well, right now, I just did a dosing study. So I'm actually studying stressed pancreatic islet cells. And so I have to actually stress them and see if they're actually stressed before I can run any of my experiments or co-culture assays. And so that's what I'm testing for today. And I'm specifically looking at stress factors, ER stress specifically, like CHOP, BIP, and ATF4, which are all pathways that get turned on when your cells are stressed. And so that is what am looking for in my cells today that I'm going to be doing with you guys.
Awesome. Okay, to back up a little bit, so you study type 1 diabetes
Yes.
and, well, all diabetes, or do you study particular type 1 diabetes? And then can you tell us a little bit about, like, what islet cells have to do with diabetes?
Yeah, and so I specifically study type 1 diabetes in our lab, but there's obviously plenty of other labs looking at type 2 diabetes. There's Modi diabetes. I have a friend that studies diabetes and Down syndrome because people with Down syndrome are more susceptible to getting diabetes. And so lots of different forms, lots of it being studied, but I study type 1 specifically. And in type 1, what happens in patients with type 1 is that their own immune system attacks their pancreatic beta cells, which is located in your pancreas. And your beta cells produce insulin. And insulin is essentially, I like to think of it like the lock and key that opens up the cells. So it's the key that has to go in and unlock the cell for glucose to enter and be utilized as energy. So if you don't have insulin, you essentially will have no energy. You won't be able to catabolize any of your glucose. And so that is a fatal condition. And that's why you would get diagnosed with type 1. And you have to take exogenous insulin injections to make up for those lost cells.
Gotcha, and so you'll have to excuse me because other than, like, a baseline knowledge of
Yeah, yeah,
of
no,
type 1
you're
diabetes
please.
and it being, like, an autoimmune condition, right? So we've learned things like the immune your immune system at some point, for some people, will attack the islet cells, the insulin-producing cells and cause this disease. Do we know more? I feel like when I learned about this coming through school, there wasn't I don't know, to me, it just maybe feels a little bit mysterious of, like, why the immune system would start attacking, that it was just sometimes it does. Do we know more now about why it does, like, when it gets triggered?
Yeah, that's a really great question. And honestly, it's pretty still unknown in the field, although there's a lot of evidence pointing towards viral infections. Obviously,
Mm.
genetic susceptibility plays a role, environment plays a role. And for each individual person, it may be a different cause. And that's what makes this disease so complicated. And finding a root cause and it being the only cause is likely not what it's going to be. And so, that's why now we're focused on creating treatments that are aimed at basically just replacing those dead cells and protecting them from those T cells that for some reason didn't like your original beta cells. So,
Are there, like, cells in these tubes right now? And can you talk a little bit about, like, you work with stem cells. So, I mean, you're creating these
yeah.
cells, which just from, like, reading a little bit about your research, like, the idea is ultimately we could use these created cells to treat to give people the cells that they need that produce insulin when their own cells aren't producing it.
Yeah, so essentially I basically work with so stem cells are a type of cell that can be turned into any type of cell in your body essentially. So a lot of people can work with like heart cells as well. They're creating beating heart tissue, which is really cool. What other things are being used? Kidney tissue, liver tissue, all these different types of fields. But specifically, we're working on creating those islets that are destroyed in your pancreas, and specifically the beta cells within the islet. And to give a little bit of background, an islet is like not just one cell, it's like a cluster of cells. So within the islet, you have your beta cells, which is what I'm focused on. Then you have alpha cells that produce a hormone called glucagon. You have stomatostatin cells, you have other types of like endocrine tissue cells in there as well. So it's not when I talk about an islet, it's talking about that cluster. When I talk about the beta cells, that's specifically talking about those insulin producing cells. And so in the lab, it is about a month and a half process to actually turn those stem cells into pancreatic islet cells that produce insulin. And so you can actually go, so we have different medias. I add different factors to basically turn different genes on and off while I'm going through this differentiation that basically drive them towards becoming those pancreatic islet cells. And so there's specific things we look for during the differentiation process. We call them like, we give them kind of funny names. We call it like lizard skin at like the end stage because it kind of looks like scales all over the cells, but that's actually like the endocrine tissue kind of forming. We have like cobblestones, so it looks like cobblestones all over. So there's little like key things that we look for throughout the differentiation. Besides analyzing them through the flow cytometer, we also can look at morphology to really see like the endocrine tissue forming into pancreatic islet cells.
Yeah, that's super cool.
Yeah,
Well, I
and
mean, so
at the
yeah,
at the end too, I can show you guys some cells we have going now and show you kind of like those golden we have golden islands, so like little islands form that are golden. So I can go show you guys some of those too at the end.
that's super cool. Well, so, I mean, for people who aren't aware, and not that I know a whole lot about this, but I mean, in terms of, like, turning any cell into a stem cell, for folks who don't know, right, there's different ways we could get stem cells in our body, right? Like, we do have reserves of stem cells in our body. People might think of, like, stem cells, like embryonic stem cells are from, you know, when you're a baby, you have more of those around, right, than not, but obviously, there's a lot of ethical concerns in the past with using anything from, a fetus, for example. So we've actually
Right.
scientists have found ways to take an adult cell, like a skin cell, right, from my arm and
Yeah.
find ways to kinda turn it back in time into a stem cell. And it wasn't that long ago that, like, the Nobel Prize went to the folks who did, like, came up with the factors that you needed to turn any cell right into a stem cell. So I feel like
Yeah.
really, yeah, how old is the stem cell field in terms of that? Like what you're able to do now? I feel like it's not
Yeah, it's
that
yeah,
old.
I would say yeah, it's pretty recent, especially in talking like science timelines, right?
Right.
Like I don't know the exact timelines, but yeah, now scientists can even take like your immune cells, like you just do a blood draw, isolate the immune cells and turn those into stem cells and use those to create your own tissue. So, for example, I have type 1, so I could take my own cells, turn them back into their pluripotent state by essentially just turning on certain genes that have been developed from that Nobel Prize, and then creating my own pancreatic islet cells and transplanting them. But in the case of type 1 diabetes, it's a little complicated because it's autoimmune. So even using my own cells could
Mm.
be a little complicated because my immune system might be like, hey, like those are the same cells we killed. We got to kill those again. So it's already programmed to kill those types of cells. So in the lab for type 1 diabetes, at least, we're thinking of ways that we can gene edit these cells and try to make them immune evasive. So avoiding the immune system. I always like to use like
Uh,
the metaphor of, if you guys know Harry Potter and he wears his invisibility cloak, but put our cells underneath that and they can hide from the immune system and produce insulin and essentially cure somebody of type 1 diabetes without them having to go on those harsh immunosuppressant drugs.
uh. Yeah, can
Yeah.
you talk a little bit about, like, yeah, what are the current, and yeah, and you just said you I didn't know this before you emailed me. I should have known, going through your Instagram, but that you yourself have type 1 diabetes. Yeah, what's that been like studying a disease that you also I mean, you probably got into the field motivated to find out more about this thing that you also have, but also, like, yeah, what are the treatments like now for people that have type 1 diabetes? And, like, you talked about the immunosuppressive drugs. Are we already at the level of doing transplants for people where they have to take these immunosuppressive drugs?
Yeah, I guess I'll kind of start. I was diagnosed when I was 10, and this was like I didn't have a continued glucose monitor where now we just wear little medical devices that constantly tell us what our blood sugar is. I did not have that when I was diagnosed, nor did like insulin pumps with these really integrative systems that give you insulin when your blood sugar is rising, stop giving you insulin when you're falling too fast. Like, we've come a long way with medical devices, but again, it's not a cure. And even I believe the science shows that a paper was published. I forget the exact statistics of it, but still, beyond besides having these great technology systems, people still suffer from those side effects of living with type 1 diabetes. So neuropathy, eye issues, kidney issues, etc. So it's not a cure, and that's why I'm here. I was told when I was 10 years old, like, hey, there's going to be a cure within 5 years. You don't have to worry about it. And well, 16 years later, and here I am. There's still not a cure, but we have come a long way. And being in this field, like working in it, I get to see it happening in front of my eyes, which is really amazing that I think not everybody gets to see and experience, but there's so many clinical trials happening right now with transplanting stem cells that are, or the pancreatic islet cells that are derived from stem cells. There's a lot of trials going on still using human donor pancreas and using an immunosuppressive drug that just isn't as harsh. There's barely any side
Mm.
effects. And so there's going to be a lot of different types of treatment options. I see it coming, and it's coming very fast. And there's also therapies right now in clinical trial that use immune evasive strategies, gene editing these cells and transplanting them. And they just kind of came out with a paper a few days ago saying that it's been successful. It's been almost 2 years, and those cells are still alive and producing insulin. So I think it's really exciting, and we're really at the cusp of something like huge.
Yeah, that's amazing just to see, yeah, like, how far it's come in a short amount of time.
It really is. Yeah, it's so exciting. And especially like myself living with type 1, I'm even like more excited about it, more passionate about it. I understand the struggles of living with type 1. And so that's why I'm here, putting in the
Yeah.
work to advance the field.
I don't remember your exact handle, but what is your handle on
Um,
Instagram?
yes, I am a PhD with T1D. So essentially my whole identity in one little tag there.
That's awesome. So, yeah, you do such a great job of, like, making science accessible, showing aspects of your life and mixing it with some of the science that you do and showing what that looks like on a daily basis. One video I saw of yours, though, that really hit home for me was, like, not long ago, you had a video where you were quite upset that some of your cells had gotten contaminated.
Yeah.
And I remember a point, probably like one of the crux points during actually it was during my master's. I ended up starting a PhD in biomedical engineering and stopping it and going into science communication instead.
Oh, wow.
But I remember being in the lab and, like, looking under the microscope, and, like, at that time, it was just like, yeah, I mean, I was dealing with human cells that I was using to test, nanoparticle delivery systems to kinda get drugs into cells, and look through the microscope, and all these little, like, worm type things were all over my
Oh my
cells, like,
gosh.
basically yeah, it was, like, a very bad contamination. I think there was a C. elegans lab, like, not far away.
Oh my
I don't know if it was
gosh.
that or something else, but anyway, they all got contaminated, and I just remember being like I mean, I think for you, described some of these assays take a month to grow these cells. I mean, that for me was just like a week of like time point delivery, having to
Yeah.
go to the lab at night and just even a week of that and finding that it was all for naught. I think that was the point at which like, you know what? Maybe science at the lab bench is not for me. Like I
Yeah.
need if I'm writing, like yes, it could be there might be a second draft, a third draft, but it's not like it's just ruined. How do you deal with I mean, yeah, how do you deal with, like, the failures that science like, that amount of, like, working at the bench where you can have such yeah, I mean, such
Yeah.
utter and abject failure.
This is such a good question, and, like, I'm still cringing at the fact that over 30,000 people watched me cry online about my cells, but that's just, like, I was like, "Do I post it?" I, like, thought about it for a long time, and I'm like, "You know what? I have to keep it real, like, this is something that's
No,
actually
it's so real.
a
I
part of
remember
my
crying so many
story."
times, yeah, in
And
the
I think,
lab.
like, so many people resonated, and everyone was, like, commenting, like, their stories about, like, "I was working on this project for 6 months, and I found out the genotype wasn't even the right genotype," and I'm like, oh my gosh, that's devastating, and, like, I honestly think getting through it is through the community I built online, is seeing everybody else being like, "I've been there, like, you're gonna get through it," has really been like, "Okay, you're right, like, this is a setback, but everything happens for a reason, and I just need to push through and try again and continue working on it until it actually works." And so I think that's kind of the mindset I've taken. I'm still upset about it, obviously, but all I can do now is try again and keep moving forward. And I think having like the type 1 diabetes community even came on there. They're like, we need you. And I'm like, you know what? You're right. I have
Yeah.
to like keep going because like we're so close. And when it does work, I'm going to get a lot of really exciting data and that I'm excited about. So
Yeah.
yeah, I think getting through that was like the online community I built really helped me, but I think also I took a day to just not think about science and I went and I painted. I do art on the side sometimes, and so I just took the day, I painted and I drew, and then I came back and I felt refreshed and I'm like, "Okay, we're going to get through this." And now I just plug forward.
Yeah, I think it's so important. I talked to, a material science PhD student on the podcast a couple weeks ago, and she had such a good answer. I mean, she was saying that, like, she just tries every time, like, something big fails to really think of it like, "It failed for a reason. What do I learn from this?" Like, I can
Yes.
actually learn more maybe from the failures than not, but, like, how she described it, she's like, "I just don't take it seriously," like, I just take it as a learning opportunity. I was like, "That is so amazing." I don't know that I can live that out. I mean, I'm a very like, I get attached, I get upset if it doesn't work, but
Yeah.
also I really like that she was saying, like, maybe to be a scientist, you kinda have to have this ability to detach, but I think the idea that, like, no, you can also be someone who cries when your cells get contaminated, and, like, that's
Yeah.
okay too, right?
Yeah, and, like, it's not like this was, like, the first time this has happened to me, like, it completely happens, just this time I was, like, so excited, and I had a huge experiment,
Right,
and they were contaminated, and it was just upsetting, but I love that perspective too, it's like, yeah, like, that is science, that literally I come in every day and it's like, "Oh," like, the undergrads ask me, like, "Why do you do this?" And I'm like, I don't know, it worked, that's why I do it. And
right,
so that's literally we're discovering new things in the lab, and, like, that's what I think's so exciting and kinda what keeps me motivated when I fall into those times, and I love it, it's like, it's not that serious, just do it again, like, it's
right,
fine.
right.
But
Yeah,
yeah,
I
there's
think
a point where it's like, I wanna graduate too, so
right. Yeah, I mean, obviously, like, there's I've also talked to some folks who do, studying samples maybe from brain tissues from individuals with a disease and taking that obviously seriously of
Yeah.
like we need to do right by the samples that we have and the data that we have from patients, and there is a whole lot of seriousness to it, but also in order to innovate and to, develop these things, you also have to have a very curious mind. You also have to be okay with like asking a question that doesn't end up being the right question and then asking a different question. And I think some aspects of, yeah, I don't know, do you have any insights in terms of like what science looks like in that way that people might not be aware
Yeah.
of?
I would say like I'm going to start with like saying like a PhD program. Your first year, you're doing kind of classes and just getting introduced to the lab. Your second year, you have that big preliminary exam where you have to go in front of your committee, which is just a group of scientists you choose to support you through your thesis and present them with like your project and what you want to do. And I will say my project now, I'm in my third year. So this was just last summer, looks completely different from what I presented, which is like crazy to think about because I'm like, I did an experiment. I'm expecting the data to look like this, but it ends up looking completely different, which makes you ask a whole new set of questions and then leads you to a whole new set of rabbit holes that you could look into and discover. And I think that's what's so exciting. You expect one thing, but sometimes you end up finding something else that isn't necessarily bad, but actually like leads to a new discovery. And if you look at a lot of Nobel Prizes, that's a lot of them, that's how they happen. Like they tried this thing expecting this, but ended up finding that it was this, and they found this new protein that is really important and could revolutionize medicine. And so I think that's what's exciting about it is that I come in every day not knowing necessarily what to expect. But I know that I have a goal of generating the data, making sure doing my assays to see if my cells are immune evasive, for example. And I think seeing that that data is working as well is really motivating and exciting during a PhD. You kind of have to find different ways to motivate yourself because like you said, like things just kind of don't turn out how you expect them most of the
Yeah.
time.
Yeah. Well, so I was looking back a little bit about in your publication record, and it looked like you had done some kind of research around genetics and even like genetics with COVID. So studying
Yeah.
like DNA, right? And then
Hmm.
obviously you've now um working a lot with proteins, right? In terms of like when you're looking at, well, you could correct me, but when you're looking at like in terms of insulin or inability to produce insulin, insulin is a protein, so you're looking at a lot of protein chemistry or dynamics or creation. Yeah, how has that changed in course of your research? How have you come to study kind of those different levels of biology?
Yeah. So I guess in my that those publications are actually from my undergrad. And so in my undergrad I did a lot more like genetics, knowing that eventually when I did go to a grad program, I wanted to study type 1 diabetes. And so
Right.
I wanted to get the skills during my undergrad to be able to do this. And so in my undergrad, I looked at patient samples a lot. So we looked at patient genetics and then they have variants. So in their genetics, they have a variant. So an A to a G, for example, instead of the normal, A that's there or C that's there. And then I would take that and test it in my cells. So I'd put that patient's like genetics into cells and then see if that affected different mechanisms of the cell. And that's kind of what I did in my undergrad. And then the pandemic happened. And so, of course, everyone's looking into COVID. And so I was able to do some
What better
computer
doing?
sciencey
I
type
like
stuff. And I'm
flying.
so bad at that type
Hmm.
of stuff. My advisor helped me a lot through that project. But really inputting code to really study the dynamics of how the SARS-CoV-2 protein interacted with the actual virus and how
Gotcha.
they binded and how strongly that binded between the different variants of COVID that existed. And so I thought that was really exciting and brought me to a different end of science that I maybe necessarily didn't ever think I would be a part of. But it's really cool to see all the different fields. And then after my undergrad, I ended up working for a year at a biotech company called Heartworks. And that's where I really saw like the stem cell field. I was like, oh, wow.
Ah,
I'm like, stem cells can become any cell. And then that's when I got thinking, I'm like, they must be doing this for type 1 diabetes. And then I looked into grad programs that were studying type 1 diabetes and stem cells and landed here. And so that's kind of like my progression of I started studying really like molecular genetics to protein science to then moving into my stem cells and then translating it to my type 1 diabetes.
That's super cool. That's amazing. Yeah, what is your in terms of, can you talk a little bit about some of the specifics of your research now? So you're actually looking at you had mentioned looking at, stressing the cells out or then looking at if they're able to be evade the immune system. Are you genetically modifying them to look for ways that we could genetically alter the cells to make them immune evasive? Yeah, like, what are the kind of key things that you're looking at in terms of what how that could apply to therapeutics?
Yeah, so the stem cells that I have, we collaborated with the lab and they helped us input these gene edits or knocked out key immune regulatory genes. So genes that
Gotcha.
are key to interacting with your T cells. And specifically, T cells are the serial killers of the immune system. So they just kill, if they bind to something, they're going to kill it because that's their job. And that's what the cells that are active in type 1 diabetes as well. And so we're identifying genes that interrupt those interactions essentially. And so we have inputted those into my cells and now I'm working to differentiate those into those pancreatic islet cells and then testing them against those T cells to see okay, are they actually interrupting those interactions? Are they able to survive when put together with active T cells? And so, what I've seen so far is super exciting. And we're also looking at other cell types because obviously the human immune system is complex. If you talk to, I'm sure you've talked to immunologists before, and it's just kind of like, crazy and you could go down so many rabbit holes, but I'm specifically looking at the T cells because those are what are part of the pathology of type 1 diabetes, and also looking at some natural killer cells and cytokines being released. So super interesting. I can't share much yet about those, but that is essentially what I'm working on.
That's super cool. Yeah, do you I mean, so is it true that you're kind of doing more of an applied, like, we think that this might be an area to therapeutics and, like, investigating that as opposed to trying to find molecular mechanisms? Like, yeah, do you f is your research a bit more, like, applied towards
Yeah, I would say mine is more applied to and translational. Like
Yeah,
eventually, like I'm hoping this could be these cells could be transplanted and cure type 1 diabetes without the use of immunosuppressants. But I also work on some other projects, some side quest projects I found that are kind of looking at what actually causes type 1 diabetes. And
gotcha.
again, like that may look different for everybody, but it's still so unknown whether it's your genetic susceptibility to viral infections. And let's
Right,
say a common virus, the Kawasaki virus, for example, enters your beta cells, and your beta cells just can't clear that virus. And so they start producing these new proteins. We call them neoantigens, and they're presented on the surface of the cell, which then brings the T cells in, and the T cells are like, we don't know what this is, so we're just going to kill it. And so, like, that's one mechanism that has been proposed, and there's evidence supporting that. But there's also evidence supporting a wide variety of other hypotheses as well. So it's like trying to really define what that is. And once we do, the pathway to a treatment can happen much faster. And so that is also a project I'm working on in the lab. And I'm using actual cells from type 1 diabetic patients, and I'm using actual immune cells from type 1 diabetic patients to really recapitulate what we're seeing inside our bodies, or in vivo, as we call it in science.
right.
Yeah.
That's super cool. Yeah, you mentioned how complex immunology is. I think of all of the fields in biology, it's
Oh,
like it's
they're all
insane.
they're
And
all
and
their
I d
own, yeah, beast.
yeah, and I don't know yeah, I haven't really studied much of immunology, but just that, there's this idea, right well, I've talked to and worked a lot with scientists who are working at the right genetic level, and I think at
Yeah.
some point, right, I don't know if this was the case when doctors were telling you, like, "We'll have a cure for it." Like, at some point in our past, as scientists were sequencing the human genome, so coming up with, like, every letter that was in, our human DNA sequence, there was this idea that, like, once we sequenced all of the DNA, basically we'd have the answers because what the DNA that's there, we can find the code of that, and that translates directly into whatever's created from the DNA, and that's it, you have the answers. And I think,
Right.
especially in immunology, from my, understanding, it's just, like, how complex it is that all that those instructions, that DNA just kinda get, like, mixed up and recombined and, like,
Yeah.
it's so much complexity added onto them. They have ways to, like, kinda jump around and create this huge complexity that is our immune system because it kinda needed to do that. Like, our immune system needs to be able to learn. It needs to be able to learn if there's a new, right, SARS
Right.
virus. We need to be able to recognize that even though that isn't in our history. So, our immune system has all of these ways to, like, kinda recombine and learn new things, which also maybe is part of what makes it so complex to study. And so when you have an autoimmune disorder, maybe that adds to the complexity of, like, treating that it's like you have to understand what's happening first, and that's just really complicated.
Yeah, and like our immune systems have memory, right? Like they remember everything that you've encountered in your life, which I think is so phenomenal, but also a part of the problem with autoimmune disorders. So, yeah, completely. It's so complicated and it's its own entire beast in itself. And I still don't think, even as a field in general in immunology, we fully understand the immune system. We understand a
Right.
lot. As I mean, I'm learning it all still, but like I think there's still a lot that can be uncovered because now we have these CAR T cell therapies are really hot right now for
Ri
cancer. And
right.
I think that's going to be really, really amazing and has already shown really amazing results. So I'm really excited to see where that goes as well.
Yeah. Yeah, actually, so there's this story, and I'm gonna mess it up, but I think it's kind of early in the kinda CAR T, but also the idea of, like, ablating, right, like, your bone marrow, basically ablating all of your blood cells to deal with, a disorder where maybe you have cancer of the blood, and it's just like, if we can get rid of all of that and basically give you a new one, then you could be cured. I
Yeah.
I have no idea. Is this an approach that's at all been tried for type 1 diabetes in terms of, like, can we just ablate your entire immune system? Obviously, that's really dangerous, but, like, isn't there some.
Yeah. I mean, there has been like talks about it. I don't think that's as much big in the field anymore because we have all these amazing trials happening right now without doing that, because you would essentially, what that does, you have to irradiate your entire immune system, so kill it all off, and you have to like retrain it. So even a common cold like could kill you in essence because, your immune system isn't prepared to take that on. Like it starts even when you're in utero and your mom, like you're starting to build that immune system when your mom gives birth, like you're building your immune system. And so I think, and this is just my opinion, like I just don't think that's the answer, but it has been done in a lot of models and it's shown to work. But is the risk outweighing the benefit in that case
Yeah,
is my question. But it
right,
is incredible that they can even like do that and study that, like, oh, let's just get rid of the immune system
right, I
and
guess it's like, yeah,
retrain
very,
it.
very, harsh approach, but I think maybe they've done that for, like, really serious, right, like, fatal,
Yeah, and no, yeah,
cases where
100%.
yeah, yeah,
I know, like, yeah, there's some, like, immune disorders too that I think they can, like, get rid of it and give them, like, a whole new immune system from somebody else, and then they have that other person's immune system, and I think that's, like, incredible because we weren't able to do that before and save those lives, so
right.
it's really interesting for sure. Or maybe, like, one day they'll make, like, some sort of immune therapy where it does just not irradiate your immune system, but like retrains it, And I
Right,
feel like
right.
that's definitely like coming for sure.
Right. Yeah, I think, today we're like, okay, we know we can just, or like cancer therapies. I mean, the very harsh ones just kind of get rid of everything that's proliferating, opposed to, like, let's target it because we
Right.
create all these other problems if we just kill everything for the for
Yeah,
the sake of, like,
exactly.
killing the one bad thing.
Right.
Well, I random aside, I had, also in going through your Instagram, I saw that you did do some pole dancing. And I only ask this because I am also an aerialist, like, I
Yes,
do.
it's so fun. It's so
So,
empowering.
have you been doing that for a while?
Yeah, I actually just had a class this week and we learned like
Nice.
I learned like a new move and I was like again, I was like awesome. Definitely wouldn't have been able to do that like 3 months ago. So, yeah, it's really exciting and it gets me like out of the lab and gets me some exercise for a couple hours and yeah, I love it.
I love it
Do
so
like
much.
what kind of dance do you do?
I
Is it
usually am
also like
doing
full?
yeah, well, I've done some pole, but I do a lot of aerial silks. So I
Okay.
started with aerial silks.
Oh,
So I
that's
do
so
mostly
pretty.
that. And I started doing some pole at the same time, but it's like just climbing, doing silks and then trying to do pole on my off days was just like too much. I ended up with some like bicep injuries. So I was like,
Ooh,
I
yeah,
can only do one of one of
gotta
these
pick one.
things. Yeah, but I love it so much. I feel like there's nothing more, like, empowering than, like, thinking
Yeah.
you can't get to the top of this thing or you're not gonna be able to climb that pole, and but with the right technique,
Yeah.
like, you like, many people that think they probably can't do it, like, can if they learn the technique. Well, maybe there's some, parallel to
Yeah. I
science
mean,
there of, like, it's all just about a learning curve.
yeah, I think it is. It's like a mix of learning curve, building your muscle as you keep trying to do it, and finally your body's like, "Okay, like you got this."
But
But
I love
oh,
it
that's so
because yeah.
cool.
Yeah. I love it just because I don't like to just work out for the sake of working out. I like to have something that's engaging my brain at the same time.
Yeah, exactly. And sometimes just like going on the treadmill or lifting is just like boring and I need something more to engage me. And so it's like such a fun and like you said, so empowering. I love it.
What do you have, I don't know, like, in current times, other than just the trials and tribulations of getting a PhD, are there things you struggle with currently or that you're working through, and, like, how do you overcome those?
Yeah. Oh, this is a deep question.
And on the other side of that, so if you can't answer that one, is like, what are you hopeful for? Like, what's giving you some heartache right now, and then what's giving you hope?
Yeah, I guess like existential crisis mode. I'm at what am I 26? Kind of having that. I'm like, do I want to do this for the rest of my life? Do I want to do a postdoc and
Yeah,
be a PI and like do that whole process, or do I want to do something else? Like, do I want to lean more into science communication? And I think, I guess what I want to like tell people is like dream big. The world is only as big as you imagine it. And so if you say like, oh, I'm not going to be able to do that, you're not going to be able to. And so always believe in yourself and dream big. Reach out to people that you don't think will answer you because you just never know what doors that will open. And like this online community has been life changing. I love it. It's opened so many doors for me that I never thought would be open. Like I get to do this really cool podcast with another scientific communicator and getting to meet so many cool people who are also doing this and really want to get the science out there and they want the public involved. I think especially right now is so important. Um yeah, and I'm really hopeful for the future. I know the future of science right now is very uncertain, but
right,
I'm hopeful it's going to be just fine and I'm hopeful that type 1 diabetes will be cured legitly within the next 5 years. At least some sort of treatment option where you don't have to live with type 1 anymore. Whether
right.
it's perfect or not, hard to say, but I'm very confident that it's very, very close. And there's a lot of really hardworking scientists trying to cure this disease and trying to cure a variety of other diseases as well. Like, I think they just came out this year with the Huntington's disease treatment. And I think, oh my gosh, that's like incredible. And so, you guys, science is still moving forward and it's amazing. I'm excited to be a part of it.
Yeah. No, I think that it there's a lot, even as a science communicator, right, that's
Yeah.
existential crisis right now, like, AI or mistrust of science is difficult right now, but at the same time, I think seeing, like, the online community around science, I mean, like, so it had been a couple of years until recently that I'd worked kinda doing more social media, management and creation around science communication. I worked for NIH for a while, and we weren't really allowed to do social media type communication for our labs, but I worked for LSU and started doing more of that and just starting to kinda get into the community and see it's changed so much, I guess, in the few years since my PhD to seeing what it is now, like, so many more scientists, I feel like now show what they're doing, like, from the lab, like, the kinda like what you do of, like, a night in the lab and creating some fun content out of it, but also showing,
Yeah.
like, that you're there and what you're doing. I feel like people are craving that, especially in the age of, like, this, AI or, like, if we don't know what's real, I think seeing real people doing real things, and
Yeah.
hearing from the experts themselves, I think is so important to people. Do you yeah, I guess, do you think about that in terms of your creation for social media? Like, what are some of your things you hope to achieve from that?
Yeah, I think you just like really echoing exactly what you said. I think people are craving like realness, like look at all the I love following all these other scientific creators as well because I love watching what they do in the lab and seeing that they're also going through similar things that I'm going through makes me feel not alone as a scientist. Where I think previously, like,
Right.
we didn't have that in this community. Like, if we felt alone, like, you kind of just had to go through that by yourself. And now you can turn to this online community where everyone's in the same boat with you and they want to support you and they want to see your science do well. And I think that's incredible and something that is new and we haven't seen before. I think like Nature just put out an article about
Yes.
scientific communication and
Mm.
I'm like, yeah, this needed to be happening a long time ago. Like communicating to the public too and like really show building trust with them so they can differentiate like what's real and what's not real. I think is so important.
Yeah.
Especially in the age of AI like you were saying, all of these like things that are coming up and I know my like mom just showed me an ad and it was like the girl that was wearing something was AI. I'm like, that's not a real person. Like, I want to see a real person wearing that item.
Right.
And so I think that's incredibly important to be able to help people identify that. And I
Yeah.
think by all these scientific creators doing this, I think is just such a big help.
Yeah. And I think that people maybe don't realize that, like, I've actually done some research in science communication about how scientists kind of presenting themselves, like, in this realness where you feel like you can grow a connection with them, right? Through
Yeah.
social media, it builds so much trust, like, and it changes people's stereotypes about what does a scientist look like? What do they do? Like, in the past,
Yeah.
there was maybe not so many options to, like, see what a science lab looked like, and most of the stock imagery it all just looks the same, like colorful tubes and white
Yeah.
coats or,
Somebody like holding a Erlenmeyer flask and looking
Yeah,
at
right.
it. Yeah.
I heard that, like, science could look so many different ways,
Yeah.
yeah, it really can change public perceptions, and I think
Yeah.
it's
I know
yeah,
science can look like somebody just sitting at a computer doing coding or
right.
analyzing a sequencing set. So I think, yeah, like you were saying, there's so many different ways that science looks. And I think people are really putting it out there too.
Do you have things you wish more people knew or myths that you'd wish to bust in, like, your field of science?
Oh, this is also a very good question. Hmm. Any myths? I think obviously science is not boring. It's not all of those things like where you're just looking at test tubes, you're labeling tubes all day. Like, me and my lab talk about all the time, like, we could have, like, a reality show for the stuff that happens in our lab. Like, I think it's very engaging, and I think we should get a reality show. Like, scientists should come together. We should, like, make a
We
reality
should.
show about life in the lab because it's chaotic, it's crazy, and in the end, as much as we complain about it, we love it, and we discover new things, and we make real change in the world, and I think that's incredible and should be showcased.
I love the idea of a reality show of science. I guess the closest I was trying to think, like, other shows with their scientists, I mean, maybe, like, Big Bang, but that had a lot of,
Yeah.
like, stereotypes too.
Yeah, like, a lot of
Uh,
misconceptions in that as well, like,
yeah.
not true, like, all of a sudden she has her PhD after, like, an exam, like, yeah. I haven't personally watched Big Bang all the way through, but
I
I've, like,
haven't either.
heard all of the, like, things that didn't quite line up.
Yeah. But yeah, you're right. I want, like like The Office, like that show, but like in a lab.
Yes, exactly. That is exactly what I'm thinking. And, maybe I have something coming like that, guys. So stay tuned.
Do you have examples maybe of like when things have been crazy fun in the lab or gone in random directions when you're not labeling tubes because it's because this is the easy thing to do while we're talking.
Yeah. Oh my gosh, there's so many stories. Well, I'm gonna bring you while I tell you a story to a
Yeah,
centrifuge
perfect.
and load the centrifuge up because that's what I have to do next. So I guess a crazy day in our lab. I'm just gonna go with what a crazy day looked for us one time. We do transplant surgeries on mice in our lab of our cells that we create.
Gotcha.
And so I lied. I gotta label these first. And so we were transplanting, I think, 14 mice, and that each transplant takes about 40 minutes to an hour. Plus, we had to take care of the stem cells. One day we had biosafety in here, and it was me and my coworker, and the biosafety person like looked at our fire hydrant and was like, "Hey, like this is expired. You got to get a new one." I was just joking around. I'm like, "Yeah, but it still works. It works."
Yeah.
And they're like, "No, it doesn't." And so I pulled it off, and the pin accidentally fell off, and it just started go. I'm like, "Oh." Yeah, so that was a crazy day. I'm not allowed in the lab during biosafety days,
Oh,
which is fine
no.
with me. Fine with me. I guess another stigma is, guys, like when I film my videos, like, and you'll see me like put a hand near my mouth, I have fresh pairs of gloves on. I didn't just touch cells and then do a video like that.
I love that
I
you had a
also,
video recently. I was wondering how you kinda come up with some of the fun ideas where you open the incubator and the cells are like, like dancing, like a Friday
yeah,
night with the
honestly, I just
cells.
hear a sound and I'm like, I just hear a sound and I'm like, oh, I could do this. Like, this would actually be good. Or like relatable or something I like people think about that like is kind of dumb. So I'm like, oh, I'm gonna just film that. And then sometimes other people relate to it. And I love it so much. I love that we all have like the same minds, really. We all think alike.
I know. Well, and I think a lot of people I mean, we spend a lot of time I was talking to someone on the podcast who studies mosquitoes, and she does a lot of, like, the
Okay.
mosquito rearing
Yeah,
and
yeah, yeah. I think I
dissection
follow
and
her as well.
yeah, fantastic, but just, like, how much I mean, she's taking care of mosquitoes all day. Obviously, these are, like she studies malaria, and so, obviously, this is a
Yeah.
scary thing. The mosquitoes in this case are not necessarily, these
Not
mosquitoes that
safe.
spread right, they're not
Yeah, not
our best
necessarily safe to
friends,
work
right?
with. Yeah.
But, I mean, I asked her, like, "Do you think of these things as pets?" And she's like, no, like, I think of them as well, she's like, "I understand that these particular mosquitoes are spreading diseases and there's issues with that," but she's like, "Yeah, I mean, I care about them, I'm taking care of them all day, I care that, like, the experiment's successful." So, like, even the cells, I mean,
Yeah.
I'm
I
sure
I
you
have
care
like
about them, you get sad if they get
yeah,
contaminated.
I'm like punishing them for dying. Like, you guys
You're
aren't
like,
dying
"Bad
because I have a
cells."
whole stock of you in my liquid nitrogen. So you guys are coming back and we're gonna get islets out of you. But yeah, I also feel motherly towards my mice when I have mice because I think the mice are so cute. Some people think they're disgusting. I think they're adorable. Um, wouldn't trade them for the world. So I like working with the mice occasionally. Every once in a while.
Yeah,
If you have too many of them, it gets to be a lot. But
yeah.
all right.
Yeah, with the mass studies, I mean, I know a lot of people, right, like and there's some moves in federal science to move towards more human models, and we're developing things like organoids, but there's still so much science that you can't figure out without an animal model, right?
Right, like, it really to truly get these cells that I'm working on into a clinical model, you have to show that it works in a live model, and so using a live animal, you can't recapitulate everything that happens in a moving body in a dish, unfortunately. We wish we
Yeah,
could, and we're trying to with organoids, but still, it's not as good as a living model, and so, yeah, it's gonna
right.
be really tough to move from that, or the FDA has to be, like less stringent on clinical requirements, right? Like if we can't use animals, well, then we are going to have to use organoid models to show that this is working, and you'll have to be okay with then moving that into a trial potentially. So I think there's like a lot of safety risks to moving out of the like live animal models.
You mentioned that you might have some cells for silica. Did you wanna
Oh,
show
yeah.
anything else before we
Yes, let me show you
wrap
the
up?
cells while they're spinning in the centrifuge. I can show
Yes.
you some cells that you have going now. And so these are at the end stage of the differentiation. So these should be endocrine tissue by now. So let me just grab my cells. Obviously, these are really important. So I don't want to contaminate them. Just gonna show you guys this little dish.
Yeah, I think stem cells are I still feel like when people say they study stem cells, it still feels, like, so futuristic to me. It still feels like,
I know.
"What
I feel
is
like when I started
that?"
working with them, I'm like, this is so cool. Now I work with them every day and I'm like, why did I do this? You know? But I
Yeah.
love it. I still love it. Because it is so like you can do so many cool things with these. Okay, I'm gonna try to show you guys. So can you see that?
Yeah.
So all of these like golden hues, these are like raised up clusters, and that is the endocrine tissue starting to form. And so these will eventually turn into those round potato looking cells, I like to call them. And they kind of do already look like that in a way, like this one specifically. But all of
Yeah.
these, like, they'll start to form like cobblestone. So like these will start to really get defined, and it'll look like cobblestone and then scaly, and then they'll turn into these cells all over when I lift them off the plate.
That's interesting to me, like, yeah, to get insulin-producing cells, do they have to have yeah, other cells types within the islet? Do they have to have those around them to really become some of the insulin-producing cells, or can you just make a whole plate of only insulin-producing cells?
So right now we can't make a whole plate of just insulin producing cells. So there are other cell types in there. So when I run the flow cytometry like I was doing today, and it was just basically me just adding stuff to tubes, is I also test for glucagon. So I look for the alpha cells. And then I also do a factor where I just look at how many endocrine cells are there. Because with stem cells, that's an issue. Like maybe we have off target cell types, and we definitely don't want that
Mm,
in our cultures. And so figuring out how to eliminate that is still a big challenge in the field that's still actively being worked on. And like getting a lot of these cells too for transplant, because they're going to require a lot of cells, getting them to function exactly like a human pancreatic islet. Those are all things that are just actively being worked on that are limitations of using stem cells right now.
gotcha. Yeah, because like you said, you're not just getting, like, maybe 100% efficiency of differentiation to only exactly what you want, so would you then need to take the cells, filter out only the ones that you want to place onto some kind of scaffold, then transplant into the patient or, the
Yeah.
mouse, like,
Yeah,
to
that's
filter you
something
need to
like, yeah, people are working on that for sure. Like basically taking out those off target cell types, but we still want to keep the cell types of the islet because, like, we know that those cells communicate with each other within the islet. So we know that's important. They need to be there. So, but getting rid of like, if we have for some reason some neuron tissue in there, we don't want
mm,
that. We want to get or neural tissue. We don't want to have liver tissue or heart
yeah,
tissue. And so getting rid of those cells is so important, and making sure there's no like stem cells that can differentiate once you transplant them, because in that case that's where those tumors form from stem
oh,
cells. And so
right,
that's part of the safety risk, is making sure we don't have those off target cell types, because those could lead to tumors. And so there are a way
gotcha,
to kind of like section out those cells in your entire population. And I think that's what most people are doing for like these clinical trials as well.
gotcha, so, yeah, I imagine, like, in terms of using stem cells for research, there's probably, like we could do a lot of it, but, like, that final stage of, like, basically cleaning it all up to be, like, 100% only what we've controlled for that to put in a patient or, or even a mouse is, like, that next level of probably a lot of extra steps and figuring out protocols.
Yeah, exactly. And like getting them to mature, like mature meaning like function just like human donor cells do. And so that's a ongoing challenge in the field, and there's a lot of work being done into how can we can we add different factors during this diff to really like get them to that point that we need them to really function how we want them to function. Where if we get them to function like human donor tissue, we don't have to transplant as many. Therefore, it's not as much work for scientists. And so I think that's actively ongoing and still a big challenge to overcome in the field. So while I think we're
Yeah.
close, we also have a
Yeah.
lot of work to do. So
Yeah. Awesome. Well, thank you so much for, yeah, showing us around the lab and explaining your science. It's been fascinating, and I definitely learned a lot.
yeah, thank you so much for having me. And I love what you're doing too. I love watching your podcast. So keep up the great work. And I'm excited to see this go live. I'm excited for everyone to see it.
Awesome. Thanks.
Thanks.